In the massive code of life, how do we find the patterns that actually matter? Welcome to BioinfoBytes, where we decode the powerful algorithms that read the book of life.
All right, today we're diving into a real heavy hitter in sequence analysis, the Smith-Waterman algorithm. You know, this thing is basically the gold standard for finding those little hidden gems of similarity buried inside a mountain of data.
So, picture this. You've got two huge DNA sequences. Most of them are totally different. But what if, buried deep inside, there's one small region that's almost identical? Maybe it's a super important protein domain. How do you find just that little island of similarity and ignore everything else? Well, that is precisely the problem local alignment was designed to solve.
Okay, to really get a handle on this, let's put it side by side with global alignment. Think of global alignment like Needleman-Wunsch as trying to line up two sequences from start to finish, the whole shebang. It's great if they're pretty similar overall, but local alignment, it's on a totally different mission. It doesn't care about the whole sequence. It's just laser focused on finding that one single best matching part, and it just ignores all the rest. Pretty cool, right?
So what's the secret? What makes this all possible? It's this one brilliant little trick called the zero floor rule. And it's so simple, it's genius. The rule is, the alignment score can never go below 0. If a mismatch or a gap would make the score negative, poof, you just set it back to 0. That's it. Now, this might sound simple, but the effect is massive. Think about it. By hitting reset every time the score goes negative, the algorithm basically says, nope, this path isn't working out. Let's forget it. So bad matches can't drag the score down into the basement. And what that means is a brand new high-scoring alignment can kick off from literally anywhere in the matrix. It's how those hidden islands of similarity get a chance to shine. So let's see how this plays out when we're actually filling the matrix. For every single box or cell, the algorithm has to make a decision. It looks at the score it would get from a diagonal move, a match or a mismatch. It looks at the score for a gap from the left or a gap from above. And then it looks at its fourth option, our secret weapon, 0. It just picks the biggest of those 4 numbers and pops it in the cell. It's that simple. Okay, the matrix is full. Now for the fun part, finding our answer. This is called the traceback. And again, Smith-Waterman does it differently. You don't start at the end like you would in global alignment. Nope. You scan the entire grid and find the one cell with the biggest number, the highest score. That's the end of your best match. Then you just follow the path backward from there until you hit a 0. And that 0, that's your starting line. Bam, there's your perfect local alignment. And believe me, this isn't just some abstract algorithm. This is a real workhorse in bioinformatics. We're talking about finding crucial protein domains that are saved or conserved across totally different species. We're hunting down tiny functional bits of code in a massive gene. It's even used to find one specific gene fragment inside an entire chromosome. This stuff is powerful. Now I know what some of you are thinking. Hey, isn't this what BLAST is for? And yeah, that's a fantastic question. Here's the difference. Smith-Waterman is the perfectionist. It is guaranteed to find the absolute best, most mathematically perfect local alignment. It's the gold standard for accuracy, but it's slow. BLAST is the speed demon. It uses some clever shortcuts to search massive databases incredibly fast, but it's an approximation. So you can think of it like this. Smith-Waterman is the detailed, careful detective, while BLAST is the lightning-fast search engine. So let's wrap it all up. At the end of the day, the Smith-Waterman algorithm is all about one thing, finding that single best patch of similarity. It's designed to find the signal in the noise. It finds that one critical island of meaning in a huge ocean of DNA. And that's why it's so incredibly important. And there you have it, the Smith-Waterman algorithm, demystified. If you want to keep digging into the amazing tools that make modern biology tick, make sure you subscribe to BioinfoBytes. Thanks for hanging out with us.